I also want to thank the organizers for what I think is a really critical topic in risk assessment and moving it forward. And I do both human and animal studies, and really I think this hit home with me when we were doing what was called the Rochester cohort study back in the early 2000s, and had moms from low SES communities coming in every 6 months with their kids looking at blood lead and IQ. And I can remember lots of times sending cabs out to find the moms, but I can also remember comments that they would make, like, you know, I don't have a job, I don't have insurance, I don't have a way to get here, and you want me to worry about lead? And I think it really sort of hit home to me, if you will, especially with respect to our animal models and with respect to public health. And as most of us know, but I think we forget all the time, most complex diseases and disorders, which is what we're supposed to worry about in public health, really arise from interactions of multiple risk factors. And in fact, chemical exposures are only one of many risk factors. And the way those risk factors come together for an individual over the course of its life really define where you are on the continuum of healthy to diseased. And they're unique to each individual as well. See if I can get this to. In contrast, most of our animal model studies, and I'm going to talk about animal model studies today largely, focus on toxicants as risk factors in isolation. So we tend to take healthy young organisms and expose them in the absence of any other context to a chemical from which we then use the information to derive risk assessments. And I'm not going to let epi studies off the hook, because all too often they focus on main effects and they don't look at interactions, not even sex interactions. So we have the same problem there. I think if you think about this, that the data that we get out of these in the absence of any context means, from a risk assessment perspective, we're picking the low-hanging fruit, and it may mean that in fact the risk levels are artifactually higher than they would be if the effects of a chemical are enhanced by co-exposures to other risk factors. So I'm going to talk about chemical exposures and stress, particularly neurotoxic metals in this talk, and why do we do that? And you heard a little bit about it from Roz, but in order to pursue this, we really needed a good biological rationale as well. Why would we study the effects of combined lead and prenatal stress? Well, first of all, they are co-occurring risk factors, as we know very well, particularly for low socioeconomic status communities. But I think as we think about cumulative risk, we also have to think about the biology. These are risk factors, that is prenatal stress and lead, that have the same biological substrates. They both affect the same brain neurotransmitter systems. They both act through the HPA axis, which you saw in Roz's talk. So they in fact share biological substrates, and they both have been shown in the human and the animal literature to produce very similar adverse CNS effects. So the logical hypothesis to emerge from that is, would they in fact produce enhanced effects when they're combined? And I want to show you a little bit of data about where we've been going with animal models to try to ask this question. As you saw in Roz's talk, this is just another depiction of the HPA axis, the hypothalamic pituitary adrenal axis. This is the system of the body that is primarily the mediator of the stress response. You can see it involves the brain, it involves adrenal, the anterior pituitary and the adrenal glands, and it has a very important negative feedback system so that after a stress response is evoked, the corticosterone, cortisol feedback, particularly into brain, but all glucocorticoid receptors to shut that response back down. And that's critical in order to return to homeostatic function. Both lead and prenatal stress affect the HPA axis. We have not been able to find in our animal models a dose or period of exposure of lead that doesn't permanently change HPA axis function. I just can't find one that doesn't, and it does it very differently by sex. They also both affect the mesocortical limbic system of the brain, which I'm just going to introduce you to briefly. It includes what's called the prefrontal cortex, the nucleus accumbens or ventral striatum, as well as hippocampus and where those cell bodies are located. And what's really important here is that HPA axis has extensive interactions with this brain mesocortical limbic system. So each system interacts with the other system, and hence some of the complexities that we see. So I want to talk a little bit about what we've seen so far with combined lead and stress, and then some of the new areas that we've been going at. And this is just a quick show of what those animal models have been. Typically, our lead exposures are initiated. These are studies in rats and mice, and we initiate that exposure 2 months prior to breeding. And the reason for doing that is because in the human environment, you don't start lead exposure when you get pregnant. It's been going on. You already have a significant body burden of lead, particularly bone, and so we try to mimic that. And we've looked at both maternal lead and continuous lead exposure across a lifetime. Not going to get into much of that. And prenatal stress regimens in rats and mice typically rely on immobilizing the animal for a period of time. We could have a lot of discussions about how that might relate to humans. In some of these studies, we give stress challenges to the offspring, and these again are restraint or exposure to cold or exposure to novelty or all of the above separated by a week. And again, I'm not going to get into those. We use different lead exposure levels, and you can see that that leaves us for each lead exposure level with 3 different groups of animals, groups that had no lead, no stress, no lead, but prenatal stress, or prenatal stress followed by offspring stress. So you can see just for this study, that's 9 groups of animals, but it's actually ×2 because we look at this separately in males and females because the outcomes are never the same, certainly in the CNS. And some of the data that I'm going to show you, we won't be looking at these stress challenges or at the high level of lead exposure. The blood lead values that I'm going to be talking about, just so you'll know, we know that blood lead values go up during pregnancy and throughout lactation. So our moms at weaning had blood leads of between 10 and 15 micrograms per deciliter. Basically, if you look at our offspring, and this is a little more complicated slide, we're talking about blood leads in the range of 6 to 10 micrograms per deciliter for our offspring in these studies. And that's in both mice and rats. So I want to introduce you to the behavioral assays that we're going to be looking at because I'm sure you're probably not familiar with them. All of the things I'm going to show you are things we do both in humans and animals. They're literally the same behavioral paradigms with species-appropriate parametric adjustments. This is one called a fixed interval schedule of food reward, and it simply rewards the first response of the animal, in this case a lever press, and you can see the child pressing a lever here, the first response that occurs after a fixed interval of time has elapsed. And in this example, after 20 seconds, the first response will produce a reward. Anything that happens before the 20 seconds has no consequence. And typically, what you'll see over time is this kind of scallop pattern of responding. That is, there's very little responding here because as you learn how long 20 seconds is, you know that reward's not available here, but as the time to reward approaches, the rate of behavior begins to increase to meet that timing exactly. And I found a little example that most people understand. This is the ERA Commons login for NIH grants, and here you can see that as the score appears on the Commons, there's very little checking of the site early on, but as the time for the score goes approaches, the rate of logging in goes way up. Just to give you an example of that. So this was, and these are just the measures that we look at. That pausing at the beginning is called the post-reinforcement pause, and then response rates are how fast the animals or humans are responding once they start responding in each of those intervals. And then we have an overall rate, which just includes all the responses divided by total session time, because I'm going to show you a couple of these over the course of my slides. So this is a relatively busy slide, but it's from that first study where we looked at maternal lead and stress in animals that had 0, 50, or 150 ppm. And the real focus of that slide is right here, this blue group. What I show you here in all of these is the 0 ppm control group, which is the black line with the dashed showing you the standard error bars, and I just show that across. You can see at the high levels of exposure, this is 150 ppm alone. We see these rate increases that we've seen before. This is 150 ppm after prenatal stress. And the blue shows what happens when you then stress the offspring. What was particularly interesting to us, we did see rate increases at these low levels, what we think of as relevant levels of lead exposure early on. But you can also see what happens when you then stress challenge these animals. You get this very significant increase in the number of responses per minute. And in fact, the 50 ppm exposure now looks like 150 ppm exposure. That was an example of the kind of thing that we were seeing behaviorally. This is another example in a mouse model of lead exposure, where we see a slightly different profile. These are the females, these are the males, again, responses per minute. And the black group is the controls, red is prenatal stress alone, green is just lead, and blue is lead plus stress. And what you see here is that there really were no effects of lead alone or prenatal stress alone in this model. It's only when you get the combined exposure. And you see a similar trend in females, although it doesn't produce conventional levels of statistical significance. If we look at those males, here again you see this data in blocks as opposed to individual sessions. You can see similarly the high levels of nuclear glucocorticoid receptors in hippocampus. Again, no real effect of lead or stress alone, only the combination. And when we do some regression, stepwise regression analysis, we can see the correlations between corticosterone levels in these animals and frontal cortex levels of dopamine, particularly metabolism of dopamine. These are some examples, again, of the kind of synergistic effects that you see, in this case with neurotransmitters. We're looking in females. Again, this is in frontal cortex. These are levels of norepinephrine. The black is all the 0 ppm, red is 50, this is 150. You can see that there is a lead-related increase in norepinephrine at 150. It really isn't enhanced any by the addition of prenatal stress or prenatal stress followed by offspring stress, except when we look at 50 ppm, where there's no effect of lead until prenatal stress is added or prenatal followed by offspring stress. And that's in catecholamine systems. These are just additional examples looking at levels of serotonin and its metabolites in frontal cortex. So again, these kinds of examples of synergistic actions. As I said before, we haven't been able to find levels of lead exposure that don't affect the HPA axis, and there we also get into the complexities of nonlinearities of exposures. Here you can see glucocorticoid levels. This is percent of basal corticosterone after we did a stress challenge in these animals, which was just simply an IP injection, which they had never had. And you get this kind of typical increase in corticosterone, and that should decline over 24 hours. What was just incredible in the males in particular, you didn't really see the initial rise. In fact, at high levels, you see a blunting, but you can see at the low levels of exposure, there's virtually no evidence of the return of glucocorticoid to low levels. So basically, the system gets turned on and it remains on. And if I add a prenatal stress challenge to that, which is this line now, you can see that it's even further enhanced. So again, you tend to get these additive or greater kinds of effects. So EPA, in their wisdom, has moved forward with cumulative risk, the national EPA, and we got funding from them and to move on with some of this literature, and we had a couple questions to address in that grant. And one of them that I thought was really important is if we're thinking about adding things like stress to cum risk assessment, I think we would want to know that its effects are generalizable to other behavioral baselines, and to other metals. And so I want to show some data from a couple, and since I only have 10 minutes left, I'm going to move through these pretty quickly. The first of these is just a baseline in which we measure learning every day in the animals, and by what we do is we have 3 levers there, and every day you have to press that series of 3 levers to get a food reward, but we change the sequence every single day, and I'm going to show you what happens in females, again, who had continuous lead exposure and stress. This is the number of responses for them required to complete each sequence and get a reward. Again, there was no real effect of lead or stress, but you can see the combined lead and stress really threw these animals for a loop. They're making 50 responses at the beginning just so it's what, how many times 3 in order to get a food reward. They do show some improvement over time, but really not much. One of the other questions that we had was, what about methylmercury, which is another neurotoxic metal, which we already knew affected the glucocorticoid receptor. It's been shown to change cortisol levels, and it also affects the brain monoamine system. So again, these are shared biological substrates of stress and methylmercury, and so one of our questions is, does this effect generalize to other neurotoxic metals? And would it therefore interact with prenatal stress? And I want to show you a very similar kind of experimental setup, different groups of methylmercury exposure. This is gestational from 2 weeks prior to breeding through postnatal day 17. This was actually borrowed from another investigator, Chris Newland, because we didn't have a model at that time, and shows you the offspring brain and blood mercury, at least in a sample we tested so far. And they've had either no stress or prenatal stress, and again, boys versus girls. So a couple of things have showed up here that were unexpected. We're still back on that FI schedule that we talked about before and how fast they're responding. You can see this is the control group. One of the things that I noticed right away is that here's the controls, here's the low dose of mercury, and here's the higher dose of mercury. So we're beginning already to see this nonlinear effect. It looks a lot like lead exposure to me. And when we look at all the groups, that is no stress and stress combined with methylmercury, here you can see the low methylmercury dose. Here you can see the higher methylmercury dose, and the open blue is the high dose of methylmercury combined with stress. In fact, it's beginning to look like the lower dose, which appears to be have greater toxicity than the higher dose, at least on this particular behavioral baseline. So clearly a suppression of behavior produced by these. What's also interesting is if you look at how long they pause before they start responding in the interval, which is the post-reinforcement pause. You can see again the controls. You can see quite clearly that there is this greater effect of combined methylmercury and stress, these open blue circles, relative to either methylmercury or stress alone. And then finally, I want to talk a little bit in my last time about one other risk factor that I think we need to think about, and that's early behavioral experience. We know that early behavioral experience is a really critical determinant of later behavior and later brain function. And in animal models, and true in epi studies as well, we almost never pay attention to this. How do we know this? Well, we know this from years of literature that you can have persistent adversity from early negative experience. This is a brain of a 3-year-old child who was neglected versus a stimulated brain, for example, in terms of adverse effects. And we can look at the literature and see the persistent positive effects of things like Head Start and some of the other early enrichment programs. They've had huge and lasting influences. Again, in our epi and our animal studies, we tend to ignore some of these effects. The same brain regions that we talked about before are associated with stress, vulnerability versus resilience. And so how does early behavioral experience potentially modify the consequences of these developmental exposures? And I want to show you, answer maybe 2 questions. Can early negative behavioral experience enhance the toxicity of lead or prenatal stress in the combination, or can positive behavioral experience potentially mitigate that? So again, another animal study I want to talk about. There is a little bit of data already in human studies showing that, of course, IQ reductions in children with increasing blood lead levels are attenuated by higher SES. And a study that came out a little while ago showing that permissive parenting, so this is low parental involvement, actually exacerbated the association of blood lead in children with scores on the McCarthy scales. So we've been trying to see how we can incorporate this into animal models, and this is the first study that we did. It was in mice, and we exposed them to lead, as I mentioned before, and then they were randomly divided essentially into two groups. One group of those mice only ever got positive behavioral experience. They were working on the fixed interval schedule and getting food reward, while some of their litter mates had what's used in the experimental literature as a negative experience. It's called a forced swim test. They're basically placed in a pool of water from which they cannot escape. So it's a stressor, although as it goes on, it's going to be a little bit less stressful. But this is a negative experience, and it occurred over the same time frame in these animals. And we then looked at lots of different outcome measures in these animals. Now, what I'm going to show you are the outcomes on brain neurochemistry for the most part. So as it turns out, that early behavioral experience is a very significant determinant of what the lead and stress effects look like. And here's one example. So we're looking at frontal cortex dopamine turnover, glutamine, which is a precursor to glutamate in the hippocampus. The red curves show you the effects of no lead, no stress, prenatal stress alone, and lead plus stress. And I only draw the lines to make it easier to see. And this is in the animals that had the forced swim, the negative experience, versus the blue line, which is the positive experience. And I think you can see that the effects are quite different. You can see the lead by stress, lead by paradigm interactions here, depending on what that early behavioral experience was. And I wouldn't say whether either of these are good or bad. These are some other examples from the hippocampus, dopamine and dopamine turnover. You can see very dramatically different effects of lead and stress in these. And we also, and I have several examples of these, we just published this, we also know that those can be directly related to behavior. For example, here we're looking at frontal cortex dopamine turnover in relation to how long they spent not swimming in the forced swim test, which goes up in a kind of linear fashion with stress, lead, and lead plus stress. And as that immobile time actually increases, you can see that frontal cortex dopamine turnover also goes down. So they're inversely related. And in the animals with the forced swim, that's in fact what we saw neurochemically. So you can in fact directly relate these to behavior. So in my closing time, I want to go to the messages, which I hope I've left you here. One of the things I think is really critical to move forward with cumulative risk is defining what risks should be studied in a cumulative format. So every time I was on the SAB at EPA for a long time, and every time I would bring this up, I would get the, oh, we have 80,000 chemicals, what do you want us to do kind of response. From my point of view, you should start thinking from the disease backwards. If we pick diseases or adverse outcomes that are clearly of economic, social consequence, we already know from the significant literature on individual chemicals which ones may play a role in those adverse health effects. So we can define what those are. So we can pick risks that are co-occurring, and I think importantly that share biological substrates. If they don't share biological substrates, it's unlikely that they're going to interact in a significant biological way or have common adverse effects. With respect to CNS effects, they almost invariably differ by sex, and yet in the epi studies and in the animal model studies, this is almost never looked at, unfortunately. I don't know if it'd be so true with other organs, but it's well known in the CNS. Prenatal stress appears to enhance the effects of at least two neurotoxic metals, lead and methylmercury. We'll also be looking at arsenic. We know it affects the HPA axis. There are lots of other endocrine disrupting chemicals that also would fit this category. And we also know that early behavioral experience is a notable modifier of the consequences of lead and stress. So the question is, how do we begin to incorporate that into our epi and our animal studies? And I think it has there are two very good reasons for doing this. One is it can provide us with mechanistic information about what's going on. We're actually in the process of doing epigenetic studies based on this right now. But secondly, it can also provide information about what kinds of behavioral experiences might provide ameliorative effects, might mitigate the effects of these early negative risk factors. So I'm going to stop at that point and again show all of the folks who've been working on this and the funding that we've had from both NIH and from EPA, and thank you very much, and again for the invitation to come and be a part of this today.